During a strong acid weak base titration, for example, titrating a weak base such as ammonia with a strong acid, hydrochloric acid, you'll be left with, at the equivalence point, all of your strong acid reacted and also all of your strong weak base reacted. You'll be left just with the salt, the conjugate acid of your weak base. So, for example, in this case, you'll be left with just ammonium and chloride. And having just these things, it's as if you've added just ammonium chloride to water. You've done calculations in which you've had to find the pH of a salt solution in which you have ammonium chloride. Now, this will give you an acidic solution. Chloride will not react with water. That would form a strong acid, which it cannot be done in the presence of water. But ammonium could react with water. It is a weak acid, and so it will produce ammonia and hydronium. And giving a higher concentration of hydronium, it will give an acidic solution. And so you should memorize that when you have a strong acid, weak base titration, you're going to have a acidic solution after you've reached the equivalence point. The pH is going to be less than 7 at the equivalence point. And so you will see that here. Initially, having just a weak base, it's not a particularly basic solution, but it becomes less and less basic and more acidic as hydrochloric acid is added, until eventually reaching the equivalence point, you have this dramatic drop-off in pH until beyond that point, the solution can become quite acidic. That is, having just this strong acid present, you can have a significantly acidic solution. Okay, so in another case, what does the titration of a polyprotic acid look like? All right, so we've actually, we do a lot of labs related to this, the facts present here. When you have a polyprotic acid, such as phosphoric acid, something that can release 2 or more protons, you will have multiple equivalence points, multiple inflection points during the titration. In this case, as you're adding sodium hydroxide to this weak acid. And so you see again that you have these 1, 2, 3 inflection points because you can release 3 protons. Now, what do you have in terms of concentrations at each compound at various points here? So initially, you have 100% phosphoric acid. Starting just with phosphoric acid, you have just that. But after you've added enough hydroxide just to remove all of the first proton, at that first equivalence point, you should have about 100% of the dihydrogen phosphate. And at the second equivalence point, you should have been able to add enough hydroxide to remove all of another proton, and so you'll be left with just monohydrogen phosphate. And then finally, at the third equivalence point, you should have added enough hydroxide to remove all of the protons from phosphoric acid, leaving just phosphate. But interestingly, you can see that halfway in between the equivalence point and the starting points, in this case, but also in this case, you have a 50-50% mixture of the original acid and its conjugate base. So where we have a certain volume of hydroxide needed to get to the first equivalence point, half of that volume gets us halfway there, so that we've only removed half of the first proton of our large number of phosphoric acids. And so we have a 50-50 mixture halfway in between these points. Now, at this 50-50 mixture, what will be the pH? Well, you know that pH can be found by having the pKa and the log of the concentration of the conjugate base over the concentration of the original acid, Henderson-Hasselbalch equation. Now, at this point, being that you have a 50-50 mixture of these two compounds, that is, they're equal in concentration, they are going to allow you to have a log of 1 there. So the concentration of the dihydrogen phosphate over phosphoric acid is 1 because they are equal in concentration, and the log of 1 is 0. And so at this half equivalence point, the pH is equal to the pKa for that first dissociation. So not only can you predict the pH, but additionally, in experiment, you can find where this half equivalence point, and knowing the pH at that point, you can find the pKa. And we'll be doing that in numerous instances in lab. Thanks for watching, guys.